This class we see about the enzyme involved in digestion. What are the enzymes are involved? What are the types of enzymes are involved for the digestion process? The enzymes are biological catalyzed produced by things, living cells. They catalyze metabolic reaction. They are soluble and colloidal substance characterized by great activity, specificity, and susceptibility to the influence of pH, temperature, and other environmental changes. The substance on which an enzyme acts is called substrate. The final product and the formed as a result of enzyme activity is called end product. So one of the example is lactose is the substrate, lactase is the enzyme, glucose, the end product is glucose and galactose. So some of the enzymes are formed of protein only, that example amylase and urease. But in most of the enzyme, in addition to protein, there is no protein group called prosthetic group, and the protein part is called apoenzyme. The enzyme composed of these two components called haloenzyme. The haloenzyme is equal to apoenzyme plus prosthetic group. The combination of apoenzyme prosthetic group is called haloenzyme. Next we see about the cymase and the zymosan. If an enzyme is secreted in the form which acts upon the substrate as such, it is called cymase. All endoenzyme are of this type. The cymase and zymosan is type of the enzyme. The enzyme secreted by active form is called zymase. The enzyme secreted in inactive form converted to active state and then is which acts upon the substrate is zymosan. But the cymase is secreted in the active form which acts upon the substrate. So the trypsin secreted in active form is activated by trypsinogen already formed the trypsin or enteropeptidase. So trypsin is secreted in an inactive form called the trypsinogen. It is transformed into active trypsin by already formed trypsin or enteropeptidase. Most of the enzymes are characterized by reversibility of their action. That is, the enzyme act in either direction. For glucose 1-phosphate is phosphoglucomutase, that is one of the enzyme that converted into glucose 1-phosphate to glucose 6-phosphate. It's a glycolysis cycle. The process will done. Then most of the enzymes are effect of temperature on enzyme work optimum temperature. The optimum temperature only the enzyme will work. The effect of pH, the optimum pH is specific unique for one particular enzymes. The effect of temperature on enzyme work optimum temperature. The effect of pH, the all enzymes are act in optimum pH. Effect of enzyme concentration, the enzyme works even when it is present low quality. Velocity of reaction increase when increase the concentration of enzyme. Substrate concentration, first substrate concentration increase, very rapid increase in the velocity of enzyme. The continuously increase, the reaction will be slowed down. Then one gene for one enzyme, the one enzyme for one substrate. Each enzyme will react only one type of substrate or group of related substrate. This property called specificity of enzyme. The specificity of enzyme is one gene for one enzyme for one substrate. The specificity of enzyme. The classification of enzyme. Classification of enzyme. The enzymes are classified 6 major classes, that is acidoreductase, transferase, hydrolase, lyase, isomerase, ligase. These are 6 major classes classification of enzyme. The enzymes are classification based on their action. First one is acidoreductase. So the acidoreductase are enzyme which involved in oxidation and reduction. The oxidation means addition of oxygen, or removal of hydrogen. The reduction means the addition of hydrogen or removal of oxygen. So the very good example for the acidoreductase is lactic dehydrogenase. That is one of the enzyme. It converts lactic acid into pyruvic acid and vice versa. The This following are the oxidoreductase, the dehydrogenase, oxidase, oxynogen. The dehydrogenase removal of hydrogen from one substrate and move to second substrate. In the equation is clearly example what are the reaction will happening AH2 plus B. So dehydrogenase enzyme, that is the A plus BH2. Next one is oxidase, the removal of hydrogen from substrate and pass it directly to oxygen. So this equation is AH2 plus half O2 is oxidase AH2O. Then oxynogenase, the catalyze the incorporation of oxygen directly into the substrate. So the next one is a transferase. A transferase is next enzyme. So the dehydrogenase, oxidase, oxynogenase, these are example for oxidoreductase enzyme. Then we see about the transferase. A transferase is a transfer a group from one substrate to another substrate. So the first thing, the equation AX plus B, the transferase enzymes will act on the substrate. Is in our end product is A plus BX. Then the hydrolase is catalyzed hydrolysis. Is directly addition of water molecule across a bond which is cleaved. It is divided into many subclasses and the nature of bound or group being hydrolyzed. Various subclasses will therefore the hydrolase, that is esterase, ethernase, peptidase, glycosidase, phosphatase, etc. Then the lysase. The lyases are enzymes which are catalyzed either the removal of a group of atom from their substrate, leaving double bonds or odd group to double bonds without hydrolysis, oxidation or reduction. Example: aldolose, enolose, fumarose, hydrolose, pyruvate, decarboxylase, etc. The catalyze either the removal of group of atom. The group of atom will be removed from their substrate, leaving double bond or add group of double bond without hydrolysis, oxidation or reduction. In the lysase, the enzyme do not do the action of hydrolysis, oxidation or reduction. The next enzyme is isomerase or mutase. This include all enzymes which catalyze isomerization, catalyze the interconversion of a compound to one of its isomers. Next enzyme is ligase or synthetase, catalyze synthesis reaction by joining two molecules. So synthetase when the name is synthesis something. So its name represents synthesis something. So the synthesis reaction by joining two molecules coupled with a breakdown of pyrophosphate bond of ADP to ATP. So the ligase or synthetase, this is the enzymes catalyze synthesis reaction by joining